And I will tell you some research we are doing on genomic prediction. I think that after the presentation of Dr. Prasanna in May, I think it's clear that CIMMYT is heavily involved in genomic selection in maize as well as wheat. There are four issues I want to talk about. I'll show you how do we use this inverse regression to attempt to improve some of the predictions that we are doing with the standard GymLab. I'll talk a little bit on genomics and pedigree-based reaction norm model, which is related to the multi-environment trial and its use in genomics and how we can use highly dimensional marker data as well as environmental covariables. Also, I'd like to explain a little bit this marker by environment interaction model, that this is a new one that we have been working in order to see if we can have a marker effect which might be overall all the environments and very specific to some environments. We've been doing this because usually has been this idea that the genomic selection models are not used, the marker effect is never estimated because there are so many markers and so few observations, so we need to have that. We have some models in which we can still estimate the marker effect and the marker effect across all the environments in one environment. And finally, a threshold model, because at least in plants, we treat all the variables as normal distributed variables, whereas in fact, disease resistances and counts, ordinal data, they need to be treated differently. And we have developed some models for ordinal categorical trait, and I'll show you some results. The main problem in genomic prediction, as we know, is that this particular matrix here, this is the response variable, these are the marker effect, these are the effect of the markers, and we know that this particular matrix is ill-conditioned. Why? Because we have, the columns of this matrix are the markers, and we might have 1 million markers, but only 100 rows, which are the observations, right? So this particular matrix is ill-conditioned. It has a very strong collinearity among the markers due to LD. Therefore, it is not full rank, not unique solution, unstable solution. Therefore, we need to seek for solutions to this big problem in statistics. And when the number of markers P is much larger than N, we can either use a shrinkage estimation and say some markers, they have an effect equal to 0. Or we use reduce the dimension of the data, reduce the dimension of the columns of that particular matrix. And this is what exactly this inverse problem approach is doing. What we are doing here is basically this. We have the response variable, which is a transformed data vector. We have this linear operator, which is the X, the markers, the vector of regression, and the vector of Gaussian error. What has been done already, so we do a singular value decomposition, a PCA, a principal component analysis on the response variable. What has been done before by De Los Campos, do the principal component analysis on the X matrix. We have done here on the Y matrix and the X matrix in order to formulate the model in a more correct manner, I would say. So we do the singular value decomposition, and so we look at the decay of the singular value, and we try to mimic the decay of the singular value using a prior distribution for the beta. And this is what I'll show you now here. Let's say this is the decay of the singular value when you do the PCA on the X matrix. The decay here is the first singular value, and then it decreases and usually takes this form here. This is the standard ordinary least square of a regression, and you see of using the singular value, and you see that at the late singular value, this is very noisy here. This is really noisy. I think that here we cannot do any prediction. So this is the decay, this is the increase of the noise. So what we've been doing is just we mimic this decay using a prior on the beta. And these are two decays for two different models that will mimic the decay of the singular value. By doing this, we believe that we might have a better prediction. So basically what we do is we make a filter or use a shrinkage estimation of the ordinary least square in order to mimic that decay. And this is the penalty that we use, using a prior in a Bayesian context, okay? And these are the results. I mean, this is not very spectacular. I mean, if you believe that we can go from a prediction of 0.4 to 0.8 or 9, that's not possible. So this is a standard Bayesian-based rich regression, and these are the two models that we use for doing this inverse problem. You see that there is, a modest increase on predictability, which might go from 1 up to even 9% for these different traits. This is female flowering in maize, well water, female flowering in stress conditions, male flowering in well water, stress conditions, grain yield, stress, well water, and these are two diseases. We see that there is an increase in predictability, and there is a decrease on the predicted mean square error, using these two particular models where we try to formulate the model in a more appropriate manner. Okay, so this is for this inverse regression. Now I'd like to talk a little bit on this reaction norm model for genomic prediction in multi-environment trials. We have big data. Why? Because we have now modern genotypes and sequencing. So we have accumulated highly dimensional markers data. That's exactly the matrix X that I showed at the beginning. So the number of markers P heavily exceeds the number of observations, so P is much larger than N. Also, we accumulate a lot of environmental data, so this also is a highly dimensional matrix that we can use in the prediction assessment that we try to do. The reaction norm model has been used a lot in animal science, and in plant science not much, but I think that this is a good solution for the genomic part, because we are using this highly dimensional marker data and highly dimensional environmental covariable data. The proposition is this: we propose a class of random effect models with the main effect of the markers and the environmental covariables and their interactions, okay? This is nothing more than a simple extension of the standard GBLAB model that uses the random effect of the model for the markers and for the environmental covariables. And actually, this model is exactly within the framework of the Cockerham and Kempthorne models in 1954, when they used the pedigree at that time. So this is the model. We have the baseline model where we have the response of the variable here in the environments and genotypes and the markers. This is the grand mean, this is the effect of the environment, the effect of the lines, and this is the interaction. What we do here is basically, let's use the genomic covariables, the markers. So we replace L, the lines, with G, which are the genomics, the markers. So this is our model. Now instead of having L, we might have G. Instead of having the environments as an index here, environment 1, 2, 3, and 4, we just use environmental covariables, and we replace E in this model with some environmental covariables that someone has collected. Okay? But also, these are the main effect model. What is interaction? Well, actually, we can do exactly the same with interaction. Instead of having the environment by line interaction as we have using the index of the environments and the index of the lines, we use environmental covariables and we use the pedigree data or the genomic data. Here, I like to say very clear, we might have pedigree data or we might have genomic data. That's the same. Well, it's not the same, but we can introduce both as a substitute of the line. Okay? And how much, now the question is how much of the G by E is explained by the interaction of the genomics or the pedigree and the environmental covariables. Well, we know that due to the imperfect LD between the alleles and the markers and the QTLs, markers may not fully account for genetic difference between the lines. We also know that environmental covariables may not fully account for the difference between the environmental conditions in different sites. Therefore, we might not expect much explanation of the G by E using genomics and environmental covariables. We have done this in several trials in maize and wheat, and we can explain the G by E not more up to 10 or 15% using genomics markers and environmental covariables. But this is something, and this is a typical example with 700 CIMMYT with lines, genotype with markers, and evaluated in 6 environments. We use here CB1 and CBU. CB1 is a cross-validation when we take only, well, not only 30% of the lines across all the environments are taken away. So in this particular case, we take the 200 lines in all 6 environments and we predict the 200 lines. And we have done this 50 times. And this, the CB2, is when we use a sort of sparse testing, where we put some lines in some environments and other lines in another environment. So this is a less difficult prediction than CB1. CB1, you predict something that you haven't seen in any of the environments. So this is the model with environments and the genomics, environment genomics and environmental covariables, environment genomics, environmental covariables, and G by environmental covariables. And here we put the full model where we use the environments, the genomics, environmental covariables, and the main effect. You see that there is an increase in prediction when we put the interaction here. We increase the prediction not only here, but also here in the CB2, which is less difficult prediction. This for this data set. I like to show this data set in maize, which comes from the testing of biparental populations. We have the 10 biparental populations that CIMMYT runs in Africa. And these are the predictions of grain yield under well water conditions, under stress conditions. We have the two prediction problems. This is one prediction problem. This is another prediction problem. We have the model, the main effect, and this is the main effect with interaction. As you can see here, we don't use any environmental covariables. We just use markers. And you see that there is a consistent increase on the prediction for each of the biparental when we increase in the model, we include in the model the G by E. Under stress, this is much more difficult. The prediction becomes, at least for some biparentals, much more difficult than in well water. In another set of wheat lines run for the Harvest Plus, using the prediction of zinc on the grain and iron on the grain. These are the prediction of using this model, the norm of reaction model for CB1 and CB2 in these 3 sites: Mexico, Oregon, which is these 2 in 2 years, here in India, the Wheat Research Institute, and in Punjab. You see that the predictions for these models where we include interaction, for some sites are relatively good, for other sites are not that good. but this is what we expect, and we like to predict things, lines within one particular environment or across environment, but this is the prediction for the lines that we set away in CV1, and the same for iron. You see that the predictions are not really bad. For some of them, or for some sites, like in Oregon, they are really good. So let's talk about this marker by environment interaction model. This particular model has the following characteristic. We have a single environment model. This is the standard McWhistle model, which is the RBLAP model, the rich regression model. These are the effect of the environment. These are the markers. This is the effect of the markers. We can put this in matrix notation. We do this. We call that this x beta equal mu u, and then we have this model, which is a standard GBLAP model. Okay? It is possible to have a model that will make a good prediction as well as be good for GWAS and for QTL detection, because that was the question many times. They say, well, no, we use a model for prediction. We don't get into GWAS or QTL. So, this is the statistical, the guy that is chasing the QTL. This might be the gene. These are the SNPs. You don't know sometimes. So is this possible? I think that this model can not solve that problem, but at least give one solution for now. The model that we have is a model that we set the main effect and the marker by environment interaction. We have the main effect of the markers across all the sites, and then we have the effect of the marker in each specific site, which is basically the interaction, because the deviation from this is this, which is the interaction marker by environment interaction. So we replace the single effect model by this model with the main effect of the markers across all sites and the effect of the markers in one of the sites. These are the results of this particular model for 3 WIT data sets, which are the candidates that Dr. Ravicin runs in CIMMYT, that they have about 700, 1,000 individuals. So here we have the 3 data set. This is the candidate that we call C45, C46, and C47. These are the environment. This is 0 irrigation bed under normal conditions. 2 irrigations bed normal conditions. 5 irrigation bed 0 tillage. 5 irrigations flat planting. These are the environments, okay? And we have 3 data sets. We have run what we call the stratified model, which is a single environment model. This is the interaction model, and this is the cross-environment model. For CV2, this is the prediction when we say, let's do a sparse testing and we have this genotype in some environments and not in others. So these are the results of the prediction models when we have this situation. Here we see that the interaction model gains accuracy on the predictions as compared with the single environment model and as compared with the cross environment. The cross environment is when you take the mean across all the environments, right? And this has been consistent across all data sets, the 3 data sets, and for each of the environment. Therefore, for this particular environment, zero irrigation, bed and bed planting, the best predicted model is the marker by environment interaction. For the CV1, it's more difficult. For the CV1, the prediction, you see there is no improvement on the interaction model as compared with the individual model. Why? Because there is no borrowing of information from from some of the individuals because they haven't been observed in any of the environments. But still the marker by environment is, on the individual side, is better than the cross-site prediction. The conclusion is that the N by E model decomposes the marker effects in genomic values. One component is stable across all the environments, main effect, and the others are environment specific. And we can plot this. I didn't do for this particular case because the markers, they have a lot of missing, but you can plot the effect of the markers in each of the environments and see how is this sort of Manhattan plot for across all the environments and for each individual environment. And usually this information is not provided by the standard multi-environment linear mixed model. The last set of slides, they talk about the threshold model for genotype, for the genomic prediction of ordinal traits. This model has been used since Janola 1983, or even before that, but in animals, not in plants. We usually, in plants, we just use the mean across all replicates, or we do a transformation. Here, we use the data as it is. These are ordinal data. The response variables, which is a disease, represent an assignment into one of the mutually exclusive categories. In this particular case, we have 5 categories. 1 indicates no infection, 2 low infection, 3 moderate, 4 high infection, and 5 complete infection. The data come from 3 sites and 300 maze lines. Therefore, this model, in a generalized list mixed model framework, is described by the distribution, the linear predictor, and the link function. I'm not going to explain this, except for this, the distribution. This is a typical multinomial, because there are 5 stages. It's a multinomial distribution, and we use the also here for this data, we use the reaction norm model that we have described before. These are the results. Now, here we cannot do the prediction. We cannot say, these are the fitted values, these are the observed, we do the prediction. No, because we have the categories 0, 1, 2, 3, 4, and 5. So we use the Breyer score for assessing the prediction accuracy. That is, the smaller, the better prediction. And you see that these are the main effects for site 1, 2, and 3. And when you put the interaction, you get about 9 to 14% increase in prediction using the G by E model. So the threshold model with G by E, they capture a seizable proportion of total variability, including the G by E. This model gives 9 to 14% over the models that include only main effects. I like to say that we do all this using the BGLR package. The BGLR package has been written by Gustavo de los Campos and Paulino since 5-6 years ago. This is a very efficient set of R codes that they can do GBLABs or pedigree. They do the Bayesian alphabet. They do the non-parametric RHSR. They might be implemented in multi-core computations. I say that because I think that the thing that the BGLR needs is a nice way to the breeders to use a Java interface. This is what it needs, because it's very difficult for a breeder to get into the R codes and run the programs. But I think that this can be used in any of the projects that we are doing. I'd like to acknowledge the researchers in the maize program, in the wheat program in Africa, India, Turkey, all over in Mexico, that generate the data for several genomic studies, as well as the research in Cornell and Kansas, and the Melinda Gates Foundation that give the opportunity for us to work together using the genotypic data and the phenotypic data, and the support that we have always in the Genetic Resources Program in CIMMYT, and as well as on the Seed Discovery Project in CIMMYT. Thank you very much. I think that's...
Thank you, Dr. Crossa, for saving some time for questions. Maybe we can have a few questions. Yeah.
Myself, Muninder from ICRISAT. Thanks for your nice presentation. Sir, I have not a question, I have a doubt, one doubt. 1 year data at 5 to 6 location is sufficient for deciding the genome selection model. 1 year data at 5 to 6 location is sufficient for deciding the genome selection model. Yeah, 1 year data at 5 to 6 location is sufficient for deciding the genome selection model.
Well, that's the data we have, but they show also data for 6 locations in different years. But yeah, I mean, that's very restricted data. 3 locations, 6 locations in 1 year. It depends where the locations are.
Sir, second question is, after defining the genome selection model, can I deploy genome selection model on double cross derived F1 population?
I'm sorry, I cannot understand.
Yes, sir. After defining the genome selection model, can I deploy genome selection model on double cross derived F1 population?
Apply the genomic selection model to
On double cross derived F1 population. Is it possible? Yeah. Yes, thank you very much.
Okay, just one more there.
Hello, sir. I'm Sanjeev. I just wanted to clarify my doubt. Like your model is explaining about marker into environment interactions, but can it also accommodate the interaction among the markers in this model only, or any other model which can calculate the interaction among the markers?
This model we don't calculate any interaction. You refer to epistasis, sort of epistasis. In this particular model, we didn't do any interaction between the markers in order to assess the possible epistasis, because in this particular set of data we have 11,000 markers. If you do the pairwise interaction, you are going to have millions of combinations. You can do that if you have only few markers like 100, 200, 1,000, even up to 2,000, but not for 11. In order to see the prediction, we can do that. That's the problem.
Okay, we'll move to next presentation, and we thank Dr. Crossa.